Hi students, good morning. So as we all know, the entire world is fighting against the coronavirus, and as a precaution measure, even our country is under complete lockdown. So under such situations, taking offline classes is not possible. So I thought to shift to online mode of teaching. And with this, from today onwards, we will be taking the online classes. So let us start our first session, and the topic for today's session is DNA replication. So in the previous classes, I have taught you the mechanism and the complete process of DNA replication in prokaryotes as well as in the eukaryotes. So quickly I will make a revision of that and then I will shift to the main topic. So the process of DNA replication involves the production of replica copies of the parental DNA, and it forms the main basis of inheritance of the genetic material. The process involves three steps: chain initiation, elongation, and the termination. Second, it is a semi-conservative in nature, means whatever new DNA is produced, it contains one parental strand and one daughter strand, making it a semi-conservative. Next, the process does not start at any point randomly, but there are specific sites which are called as ORI sites from which the DNA replication process starts, and it is usually bidirectional, moving in both the direction from the point of origin. The process is template dependent as well as primer dependent. So template is the parental strands. Both the strands act as a template and synthesize the new strand. So without the existing strand, we cannot synthesize the new strands in VO condition. Then the primers provide the free 3' OH group, which is the main requirement for the enzyme DNA polymerase. And then the last and the main feature is the process is not continuous on both the strands. So on one strand, DNA is synthesized in a continuous manner, that we call it as a leading strand, and on another strand, DNA is synthesized in the shorter fragments. So one strand is leading, another is a lagging strand. So leading strand, only one primer is sufficient, whereas on the lagging strand we need many different primers. So these are the few important features of DNA replication. Next we shift to the differences or the proteins involved in prokaryotes and eukaryotes which carry out the each step. So the mechanism is same in both the organisms, but the proteins involved in carrying out each step makes the process different in prokaryotes and the eukaryotes. So the first feature is origin of replication. So if we see ORI site in prokaryotes, there is only one ORI site which is of 245 base pairs, whereas in eukaryotes there are many ORI sites and they are usually less than 200 base pairs. When we see the structure of origin of replication, it is called as ORIC site, particularly in E. coli, and it is made up of two different structures. That is, one is 9-mer sequence which is repeated 5 times and that forms the binding domain, and the another region is of 13 base pair sites and it is repeated 3 times. From this region, the actual melting or the opening of two DNA strands starts. When we come to eukaryotes, that is in the yeast, the ORI site is usually called as autonomously replicating sequence or ARI sequence, and it is made up of 4 different regions: A, B1, B2 and B3. So A and B1 sites provide for the binding, and B2 is the main site from which the replication starts, and B3 also, that is autonomously binding factor 1 will bind to the B3 to maintain the stability of that process. Second feature is recognition. So these ORI sites are recognized by different set of proteins in both prokaryotes and eukaryote. In prokaryote we call it as a DNA A protein, and in eukaryote it is CDC6 protein, which is activated or which binds at the S phase of the cell cycle. Then comes the after recognition, we need to separate this two strand, and that is done by the enzyme helicase. So helicases break the hydrogen bond between the two strand. So helicase in prokaryotes is encoded by DNA B gene, and here it is called as MCM helicase. Now, once we open the two strand, they still have the tendency to reform the bonds, and to make them or to keep them separate, we use some proteins called as single stranded binding proteins. In prokaryote, we call them as single stranded binding proteins, whereas in eukaryotes, it is replication protein A. Once we have separated the strand, our next step is synthesis of the primase. Synthesis of the primer, which is carried out by the enzyme primase. So primase in prokaryotes is DNA G, whereas in eukaryotes that function is done by polymerase alpha itself. Now, once we synthesize the primer will provide the free 3-OH group. Our next step is elongating the primer. So that elongation is done by the enzyme polymerase 3, and in prokaryote the elongation on both the strand, that is leading as well as lagging, is done by DNA polymerase 3 only, but in the eukaryotes the leading strand is done by polymerase epsilon and the lagging strand is elongated by polymerase delta. So here two different enzymes are involved in replicating the two different strands, epsilon and delta. Then when we start reopening the strand at particular point, the other strands get extra coiling, which we call it as a supercoiling. So to avoid that supercoiling, there is a function of another enzyme called as topoisomerase. So the word itself tip topo, in the sense it is going to maintain the topology or affect the topology of the DNA. So in prokaryotes that is type 2 topoisomerase, which is usually gyrase A and B, whereas in eukaryotes both type 1 and type 2 topoisomerases are involved. Then the processivity, or how many nucleotides are added per second, that is has to be maintained, and the processive component in prokaryotes is beta clam, whereas in eukaryote it is proliferating cell nuclear antigen PCNA. So these two are required to maintain the processivity, and these processivity components are loaded by gamma complex in prokaryotes, whereas replication factor C will load the processivity component, that is proliferating cell nuclear antigen, in case of eukaryotes. Once we do this, DNA polymerases will go on replicating the strand, and when we reach the end, that is recognized by the TUS protein, and the process will stop. Polymerases cannot extend further, so DNA polymerase 3 and delta and epsilon will be released from that site. Now when we have completed the process, whatever RNA primers are there, they has to be removed, and that is done by DNA polymerase 1 in case of prokaryotes, whereas no eukaryotic polymerase enzyme is having that activity to remove the primer, whereas here two new enzymes come, which is called as RNase H, which degrades the RNA from a RNA DNA hybrid, and then the flap endonuclease 1, which will remove the last base of that primer. Now, if we see the processivity, it is usually 100 base pairs per second are added on per fork, whereas when we come to eukaryotes, it is simply, sorry, in prokaryotes it is 1000 base pair per second per fork, whereas in the eukaryotes it is 100 base pairs per second per fork. So if you see, the speed or the processivity is more in prokaryotes compared to the eukaryote, or the process is faster. but the number of ORI sites are more in eukaryotes than in the prokaryote. Obviously, eukaryotic genome is larger in size, more complex, and hence the process must be more precise as well as accurate. So, to maintain that accuracy, the speed or the processivity is also less. So with this, we have finished the features in both prokaryotes and eukaryotes. Now let us move to our main remaining topic, that is replicating the ends of chromosome. So as we know, the nature of DNA in prokaryote is circular. So there the problem of end does not arises, whereas it is linear in case of eukaryote. So the parental chromosome produces two DNA molecules and each of them has an RNA primer at the end. So once we remove the RNA primer, right, so you can see here, this is a double stranded DNA. We have completed the process, we have separated the two strand, this one strand and this one strand, and once the process gets finished, if you see here, after removing this primer and this primer, a gap will be created on both the ends of the both the strands towards the 3' OH group. Now, we all know that RNA polymerases can extend this strand only if they have the free 3' OH group. But now after removal of the primer, we are not having any free 3' OH group here, but at both the ends we have got the 5' phosphate group. So RNA polymerases cannot extend this strand. So what happens? The spaces or the gaps are left at it as it is, but if we leave these gaps as it is and if we combine them, after every round of replication, some of the information will be lost because this much of, for example, here three nucleotides are not generated in the daughter strand, right? So three nucleotides are lost means one codon is lost. One codon in the sense one amino acid is lost, and that produces the major effect in the future generation. So this should not happen and we should restore all the information, right? So how, what is the solution for that problem? So this problem is solved by two elements. First element is our enzyme telomerase, right? And the second element is the telomere ends. So if we know the telomere ends, telomere which is called as the ends of the DNA are having repetitive sequences, right, which do not code for any of the gene, and these repetitive sequences are conserved in all organisms from E. coli till the human beings, and those are unique in the sense in each organism the telomeric and repetitive sequences are unique, and those sequences are recognized by the enzyme called as telomerase. For example, I have given here one example. The sequence of telomere in the human beings is 5' to 3' TTAGGG, and such 100 to 1000 times it is repeated. So this at the end, if we do the end sequencing, this sequence, that is a hexamer, is repeated nearly 100 to 1000 times to maintain the integrity of the actual end of the chromosome. Now what is the enzyme? Telomerase. So telomerase maintains the chromosome length by adding telomere repeats to one strand, right, with the end. In the end, it will go on adding the new repeats, which serves as a template on the previous DNA replication at each end of the linear chromosome. I'll show you the diagram, then you will understand what actually it means. Now, telomerase is a unique enzyme which is made up of both protein component as well as the RNA component. All our, most of our enzymes are protein in nature. There are few enzymes which are also RNases, that is RNA in nature, but this telomerase is made up of both protein and the RNA. Now the protein part of this will do the catalytic activity, that is it will perform the polymerization activity, and this RNA will provide the primer. So our both problems are solved. We also got the activity of polymerization, which is provided by protein component, and the activity, that is the primer, which is provided by this RNA, which is there in the telomerase itself. So now if you see here, it is utilizing RNA to produce the DNA, and that process we call it as a reverse transcription, right? So synthesis of the DNA from the RNA. Transcription is synthesis of RNA from the DNA, but here we are synthesizing DNA from RNA, and that's why it is called as reverse transcription. So telomerase is example for reverse transcription, and as it is utilizing the RNA, it is also called as RNA dependent DNA polymerase. Utilizing RNA, that's why RNA dependent, but synthesizing DNA, that's why DNA polymerase. So let us understand this process. So before that, I would like to show you some conserved sequences in different organisms. So first one is in Arabidopsis, we have the sequence TTA-GGG. In human beings, it is TTA-GGG. In so on, other organisms are also having, and this is the structure of the enzyme telomerase. So if you see, this is the protein component, and this 5' to 3' or 3' to 5' our RNA. So if you see, this is a human telomerase enzyme. The repeat of human is TTA-GGG, and this repeat is complementary to this region, that is AAUCC. So there telomere will form the complementary binding with telomeric end of the parental DNA, and then it will synthesize the new strand on the parental DNA itself. So understand this process. So if you see, chromosome ends after primer removal. So this we have removed the primer from the, this is your parental strand and this is your daughter strand, and here there was a primer. So we have removed the last primer. Now we are having here 5' phosphate group, and we cannot synthesize this strand. Now what telomerase will do is, it will attach at this site. So if you see here, TTAGG was the conserved telomeric sequence in human beings, as seen on the last slide. Now if you see the telomerase enzyme, its RNA is having certain bases which are complementary to the parental DNA, so it will form the base pairing. Now if you see, the parental strand is having the free 3' OH group, so we can extend this strand, and to extend this strand we are also having the complementary template strand, right? Here there in this figure 3' OH group was there over here, but no complementary template strand over here, right? So why we need both template as well as the 3' OH group, and both the requirements are fulfilled by this telomerase. So once it binds to the parental strand, it will add new nucleotides here. So if you see, opposite to C, G will be added, you can see here. Opposite to A, T will be added, T, then A and G. So this much amount of strand will be extended. Now pay attention, actual strand in the parent was up to this only. Now whatever these nucleotides are added, they are extra nucleotides, right? So such 4 5, so up to 10 times such sequences will be repeated. Now if you see the second slide, actually this was our parental gene region. Now once it has synthesized this new strand, this telomerase will shift forward. So it has, from this location it has moved to the this location, and again it is forming complementary base pairing with newly synthesized strands, and again extending the parental strand towards the 3' end. Understood? So it will do it until some 100 to 150 new bases are added. So our actual end was TTAGGG up to this, and this all ends are newly synthesized, right? Now once it completes this, the telomerase will leave this site. Once it adds some 100 base pair, it will leave that site, and after that a new primer will be. So we are having this much amount of the template strand. Now RNA primase will once again synthesize the primer over here. Okay, that is 5-2. Here there will be 1 minute. Here there will be 5' phosphate group, and here there will be 3' OH group, right? 3' OH group on the new strand. So we can extend this strand in this direction, and we can reach up to the our actual end. So we have synthesized, you can see here, this much stretch of the new DNA, but still this DNA will be overhanging only. But what we have done is, using telomerase, whatever actual DNA sequence was there in the parental strand, that we have replicated in the daughter strand. So now this was your extra information. So what happens is, this, it is extra information, and as I told, these telomeric ends do not code for any of the proteins. So what is their function? So if you see, most of them are G rich, that is guanine rich, and because of this nature they protect the chromosome, the other parts of the chromosome. So the telomeric ends are GC rich, and they protect the telomeric ends, right? So in most of the organism, this single stranded overhang, which is G rich, will bend on itself and get inserted into the daughter strand, forming a loop like structure, which we call it as a T loop or the D loop. So this two, that is the overhanging sequence, will bend on itself, it will enter into the daughter strand, and it will form the loop. So it is called as T loop or termination loop, and that will protect the main sequence of the chromosome. So the telomerase saves our information from getting lost generation to generation. Now most of the cell DNA polymerase loops back on itself, that what we read. If we see in mammals, telomerase activity is limited to the immortal cells. Immortal cells, which are immortal cells? Which are always growing, stem cells, germline cells, and the main importance are tumor cells. This is the main reason why tumor cells are immortal, or why we cannot control their division, is they have highest activity of the telomerase. So information is not at all lost, and as they have lost the control over cell division, they keep on growing. But if you see, the absence of telomerase activity in the cells results in progressive shortening of the chromosome. So if there was no telomerase activity, after each round of DNA replication, some amount of nucleotides are lost. In turn, some codons are lost, and that leads to production of incomplete proteins. And if we don't produce a functional protein, most of the cellular processes will be hampered, right, thereby limiting the number of cell division. That's why we say that after certain period our growth stops, our or the cell loses the capacity of division that is occurring in our normal cell. But if you see the tumor cells, they are having highest rate of telomerase activity, and that is also one of the main reason why finding or curing those cells is very difficult. So that was how we are going to replicate and maintain the ends of enzyme. So I will revise once again. That process is carried out by two enzymes, telomerase, which is behaving as enzyme as well as RNA. So it will provide a template as well as a primer, and in that way we can restore the parental DNA information. Now the next step is, we have, okay fine, we have finished the replication, we have multiplied the end also. Next is, we know that DNA is wrapped with the histone proteins, that is in the chromosomes the DNA is bounded with the histone proteins to form a nucleosome, and finally these many nucleosomes come together to form a solenoid, and then it will form the chromatin fibers, and after further condensation it will take the forms of chromosomes. So how, so replicating histone is also very important, and usually histone replication starts in the S phase itself soon after DNA replication starts. So you can see here, this was your parental nucleosome, parental DNA, and here as the new strands are produced, the DNA replication have also started and the coiling or the condensation has started. So histone proteins, which provide the structural framework to our DNA, play very important role in the condensation. So there are 5 types of histone. One is missing here, that is H1, H2A, H2B, H3 and H4. So this H1 is acting as a linker between the two octameric units, whereas H2A, H2B, H3 and H4 together combine to form a octamer. So 2 unit of each of this combine together forming the octamer, and around that octamer nearly 146 base pair of DNA combines forming a nucleosome. So you can see here, as soon as the parental DNA has replicated and daughter DNA is produced, the histones are also getting synthesized and they are forming the octamers, and the DNA is getting coiled around them by, and finally it will assume the nucleosome conformation, and at the end it will form the chromatin fiber. So with the end of nucleosome assembly, the process of DNA replication comes to end, because nucleosomes are also very important in maintaining the integrity of eukaryotic chromosome. This process we does not see in the prokaryotes. So with this we have finished today's session. We have seen the process of DNA replication and also how the ends of the eukaryotic chromosomes are maintained and how the nucleosome is assembled. So here is homework for you people. You can solve this question. I have given the some of the processes and the enzymes here in detail. I have taught you this all enzymes in the class, so just try to match these and send me your answers in the. So thank you, keep learning and keep growing. I hope you will enjoy this session. Once again, thank you, have a good day.